Good morning, everyone. We are going to finish our chapter 3 material today, continue looking at energy, the requirement for energy, the consumption of energy in biochemical reactions. We'll start today with looking at one of the roles of ATP, doing the work of the cell, driving some non-spontaneous reactions. Reminder that we will have our second exam a week from Monday. So one more chapter's worth of material next week. We'll be covering cell respiration in chapters 13 and 14. And for that reason, Joshua is holding an extra office hour next week. So he always has his Thursday morning, 11 o'clock office hours, and the week before each exam, he will also have an extra office hour. And so you can put that into your calendar. Tuesday at 2 o'clock will be an extra one as well. I recommend that people find some time in the next few days to catch up with all of the material that we've been covering. Make sure that you feel comfortable with material from chapter 9 and membranes 11 and 12, and our energy processing in chapter 3, so that you're ready to continue and learn the next week's chapter of material. So you're ready for exam 2 a week from Monday. Remind everyone to make sure that you have signed into your Reef account, that you have joined our course. And I thought that today we would make this a bonus clicker day. I haven't done that in a while. So today's points will be added on to the top of your accumulated points. So today's points will be out of 0, all right? So let's take a look at where we left off on Wednesday. We described the use, or how the cell uses ATP, an energetically favorable reaction, hydrolysis of ATP, which is illustrated over here on the right, this large drop in free energy from ATP to ADP to drive a non-spontaneous reaction, right? That's one of the one of the types of work that a cell will undertake. And so let's look at an example of how ATP hydrolysis can actually make an unfavorable reaction like this here from X to Y that requires energy, how to make that reaction move forward. So here's our energetically unfavorable reaction. X converted to Y. Y has more free energy than X does. So that in that direction, that's an unfavorable reaction. Well, as we'll see, what really happens is that conversion of X to Y occurs in 2 steps. In the presence of ATP, X will be phosphorylated. A phosphate group will be added to the X, which makes it more unstable. So our phosphorylated X is what we call a high energy intermediate in this process of converting X to Y. And then the cell will undertake the next step, in which this phosphorylated, this unstable, chemically unstable high energy intermediate phosphorylated X is converted to Y. In that case, that phosphorylated X converted to Y, the phosphate is released as an inorganic phosphate. So that overall, when these 2 reactions both occur in the presence of ATP hydrolyzed, X is converted to Y. So overall, X to Y is favorable, right? It will proceed in the forward direction. So let's look at an example of how coupling ATP hydrolysis to a energetically unfavorable reaction can drive it forward. In this case, we're looking at a condensation reaction at the bottom here, joining two subunits together to make a more complex, let's say the beginning of a polymer. A condensation reaction, a dehydration synthesis, is non-spontaneous. It's not favored. Energy is required in order for A to be joined to B to form this more complex molecule. And so what we see is that there are two steps. The activation step is in the presence of ATP. ATP is hydrolyzed to produce ADP, and that terminal phosphate is added or linked to subunit B. So now we have a phosphorylated compound B, a high energy intermediate, which because it has so much free energy and it is now chemically unstable, it is much easier for a phosphorylated B to exchange its phosphate for subunit A in this condensation step. Replacing the phosphate with A will allow the joining of A and B to form our polymer. So let's look at a more specific example of a condensation reaction. Condensation, A plus B forming simple molecule plus simple molecule being joined together, a new covalent bond forming something more complex. As a specific example, we can look at conversion of one amino acid to another amino acid, glutamic acid to glutamine. Glutamic acid is an amino acid, but it can be converted to glutamine in the presence of ammonia. An amine group added to glutamic acid forms the amino acid glutamine. Right? This condensation reaction, this dehydration synthesis is non-spontaneous. It's going to require an input of energy. So let's see where ATP hydrolysis comes into this. Same sort of thing we were just looking at. Here's our glutamic acid over here. This is an amino acid, right? There's the amino group, there's the carboxyl group. Here's the side chain. This is a polar amino acid. And we're looking at this hydroxyl here. In the presence of ATP, in the activation step, ATP will be hydrolyzed, releasing ADP, and that terminal phosphate will form a new covalent bond right here at that hydroxyl group on that carbon to phosphorylate it. So we have this high energy intermediate, a phosphorylated glutamic acid. Right? So in our first step, our activation step, ATP hydrolysis results in transferring of a phosphate group to the glutamic acid. Now we have our high energy intermediate, more unstable, more chemically reactive, and that more chemically reactive high energy intermediate is now more likely to be able to interact with ammonia and replace the phosphate group on this carbon with an amine group. In our second step, our condensation step, exchanging the amino portion of the ammonia for the phosphate on the high energy intermediate. And we end up with our glutamine. Right? Replace the hydroxyl on that carbon with an amine group via the phosphorylation step. Overall, glutamic acid to glutamine was an energy requiring process. It's non-spontaneous. It could only occur by being coupled to, this condensation step was coupled to the hydrolysis of ATP. And the reason that this can move forward is because the hydrolysis of ATP releases more energy from ATP converted to ADP plus the inorganic phosphate. That reaction releases more energy than the condensation of glutamic acid to glutamine consumes. So we have a net overall release of free energy. So overall, this glutamic acid to glutamine condensation is favored as long as that reaction is coupled with hydrolysis of ATP. All right, and we're going to see lots of examples of this. We'll see examples of multiple types of biochemical pathways where one step involves the phosphorylation of a substrate. That phosphorylation step creates a high energy intermediate, which can then be converted to the next product in the reaction. That driving that non-spontaneous overall reaction requires ATP, requires the ATP hydrolysis. So let's look at a different type of an example. Here, excuse me, in this question, we have two reactions. We have reaction 1, which has a delta G of minus 100 kilocalories per mole. That's the change in free energy. And we have reaction 2, Y converted to B, that has a change in free energy of 50 kilocalories per mole. So what can we say about those reactions? And notice what I'm asking you to identify is which of those 4 statements is false. How are we doing? Everybody have a chance to vote? Remember, you're voting, you're selecting which of the statements is false. All right, let me close it up. And it looks like the popular answer is the correct answer. D is the one statement that is false. So the other's correct. Reaction 1 is favorable because we have a large negative change in free energy. And that is due to the hydrolysis of ATP. Reaction 2 is an example of an energetically unfavorable reaction, right? Because the change in free energy from Y to B is positive. B has more free energy than Y does. If these two reactions are coupled, then reaction 2 can proceed in the forward direction. Right? And what we're seeing is that hydrolysis of ATP will phosphorylate Y, convert it to a high energy intermediate, and then in the condensation step, Y can be converted to B. So A, B, and C are correct, but reaction 2 can't drive 1 in the reverse reaction. It wouldn't do that. And even if we had reaction 2 in the reverse reaction from B to Y, there's not enough change in free energy to be able to drive 1 in the reverse. So for a couple of reasons, D is incorrect. So ATP is an important energy carrier, a molecule of usable energy in the cell, ready to use energy. Of course, we take in our cells into our body and into our cells other high energy, complex, high free energy molecule. Those macromolecules like polysaccharides and polypeptides and fats are high in free energy because they're storing energy in their chemical bonds, right? The potential energy in their chemical bonds. But that stored energy can't be used to do any work of the cell unless that energy is transferred, extracted from those macromolecules and transferred to the energy carrier, the mobile ready to use energy carrier ATP. And that's what we are looking at in our next chapter when we follow the different steps of cell respiration, the multiple processes really that are involved in cell respiration, the way that a cell will harvest the energy that's stored in macromolecules and transfer that energy to small carrier molecules, ATP plus temporarily other small molecules that we'll introduce today. So if we think about metabolism, the way that a cell manages all of its matter resources, all of its chemicals, all of its energy resources, we are looking at the intersection really of catabolic pathways, breakdown pathways, and anabolic pathways, synthesis pathways. Our cells take in our food molecules, our complex high energy molecules, and a cell will be breaking down through various catabolic pathways, break down those macromolecules into smaller components. Those smaller components, and of course this is spontaneous, more complex to less complex, more disorder, but all of those individual smaller, simpler components can be converted to higher energy, more complex macromolecules that the cell needs via a variety of anabolic pathways, synthesis pathways. But these are non-spontaneous reactions. They require energy. So the energy that's released from the catabolic pathways, such as ATP, are used to drive these non-spontaneous reactions to synthesize the macromolecules the cell needs. Again, these catabolic pathways, not all of the energy of those molecules are converted to useful forms of energy like ATP. Some of the energy is lost as heat, right? The lowest form of energy, more disorder. But that heat is actually used by the organism. That heat helps keep our body at body temperature, 37 degrees Celsius. Some of that heat energy is used to help a reaction overcome its energy barrier, its activation energy barrier. In some cases, that heat is enough, and maybe along with an enzyme directing it, catalyzing it, some of that heat is used to actually allow a chemical reaction to move in the forward direction. So another way of looking at these metabolic pathways is considering, over here on the left-hand side, we're looking at our catabolic reactions. Again, in the context of energy use or energy release, energetically favorable reaction, breaking down of a food molecule. That food molecule is oxidized. Energy is extracted from that fuel molecule and carried in our ATP, right? Energy is used to convert an ADP to an ATP, right? To add that extra phosphate to the end of the ATP molecule, high energy molecule. And the ATP is the energy that when that ATP is hydrolyzed back to ADP, that is used to drive an energetically unfavorable reaction, a non-spontaneous reaction, a building reaction, synthesis reaction to make a high energy molecule that the cell needs. Right? So we're looking at energy moving within a cell from one molecule to another, comes in as fuel or food, and some of that energy is used to build synthesize other molecules that the cell needs. That's one piece of the work, one type of work that the ATP does in a cell. Other work is transport work, right? ATP required to drive solutes against a concentration gradient. We'll see later ATP is used for mechanical work to cause molecules to move relative to each other in a cell. So let us take a peek then at the source of the energy. The original source of energy in a living cell is light energy from the sun. So what we're asking here then is, how does that energy enter a cell? Sunlight, in the process of photosynthesis, that energy is used to create or to form high energy molecules in the cell. High energy molecules like sugars from the reduction of atmospheric carbon dioxide. So carbons of our sugars enter the cell as carbon dioxide. The carbons of the carbon dioxide are reduced to form high energy molecules. In the process, water is split and H2O is oxidized, and the oxygen is released in the process of photosynthesis. And we'll talk about that 2 weeks from now. The energy of sunlight that has built these high energy molecules from atmospheric carbon dioxide, those high energy sugars and other organic molecules can then be broken down in a cell, whether it's a plant cell, an animal cell, a protist, to a prokaryote, to be able to extract the energy of those high energy molecules into useful chemical bonds like the bonds in ATP. In that process, the carbons, the high energy, high free energy in the carbons of the sugars, those carbons are going to be oxidized. Oxidation allows for the release of energy. And so the carbon is then converted back to carbon of carbon dioxide that's released from the cell. This process requires oxygen, and that's what we're going to be talking about in the next chapter in cell respiration, overall releasing water. So the energy is coming into our cells. The ultimate source of energy is sunlight. That energy is captured to produce high energy molecules, which then by catabolic pathways are broken down, and that energy is then transferred to useful chemical bonds, such as ATP. So we say that sugars are oxidized. In the presence of oxygen, sugars are broken down. And we want to take a look at how that occurs in a cell relative to in a non-living chemical reaction, a chemical reaction outside of a cell. If sugar is oxidized, sugar is burned, degraded, broken down in the presence of oxygen in a single step process. If we're looking at the energy profile of sugar, a high free energy molecule in the presence of oxygen, can almost instantaneously, as long as that activation energy is overcome by a spark or heat from a fire, will almost instantaneously be converted to carbon dioxide and water. All of the energy that was held in that sugar molecule is released as heat, as light, right? That's combustion, right? Breaking it down immediately. Drop in free energy, high energy molecule sugar, low energy carbon dioxide and water. The sugar is oxidized, the oxygen is reduced. But what a cell does is, instead of directly burning the sugar, it's going to oxidize the sugar in a number of very small steps. Slow, controlled, enzymatically catalyzed breaking down of that sugar, releasing the energy in small steps so that the energy can then be stored in carrier molecules. This also minimizes the energy that's wasted or lost as thermal energy that will just be dissipated from the cell. So if we look at that same energy profile and the oxidation of a sugar in a cell, we will have an overall same change in free energy, about what, 600 kilocalories per mole, a big change, a big drop in free energy from the free energy in a sugar molecule as it is oxidized to carbon dioxide. But this is a regulated enzyme catalyzed series of drops in free energy. Small activation energies overcome by body temperature, overcome by specific enzymes catalyzing specific chemical reactions in small steps, extracting the energy from that sugar molecule and eventually using that energy to synthesize ATP, that useful mobile form of energy. So that's what we're going to be looking at in cell respiration, how a cell can extract energy into useful forms from its fuel molecules, such as a sugar molecule. Overall, sugars oxidize to carbon dioxide. The oxygen, let's go back for a second, the oxygen is reduced to water. So what do we mean by oxidation? Oxidation is when a molecule loses electrons. This occurs in the presence of oxygen. That's what we call oxidation. But the atom or a molecule is losing electrons. This is an energetically favorable reaction. Energy is released. This has to be coupled with a reduction in which another molecule gains those electrons. That on its own is an energetically unfavorable reaction. It requires energy. And we're always going to see oxidation reactions and reductions coupled with each other. Somebody's got to gain the electrons, taking electrons from another molecule. So let's look at an example of an oxidation and reduction or redox reaction. If we just look at simple atoms, and here, let's say that atom 1 is a carbon atom and atom 2 is an oxygen. When a bond, a covalent bond, is formed between a carbon and an oxygen, remember oxygen is highly electronegative. A pair of electrons will be shared between those two atoms. That's a covalent bond, right? Share a pair of electrons is a covalent bond. That's the attraction between these two atoms. But if one of the atoms is much more electronegative than the other, like oxygen is more electronegative than carbon, the shared electrons will spend more of their time around the more electronegative atom like oxygen. And so oxygen becomes partially negative, it becomes reduced, right? Its positive charge is reduced. That's what we call it, reduced. It becomes more negative, partially negative. And the other atom, in this case, the carbon, has partially lost its electrons because they're spending more time around the oxygen than the carbon, becomes partially positive. So one atom is oxidized and the other is reduced. Right? A polar covalent bond. The electrons that are being shared are not shared equally. They're spending more time around one atom than the other. And we can see an example of that if we're looking at, and what we're really going to be focusing on is oxidations of carbons in this process of breaking down this carbon-based fuel molecule. So let's look at a type of fuel, methane. Methane is a carbon that shares 4 pairs of electrons with 4 hydrogen atoms. Right? Those are all non-polar covalent bonds. All of the electrons are shared equally between the carbon and each of those 4, its valence electrons, right? In those 4 non-polar covalent bonds. In the presence of oxygen, that carbon can be oxidized. This carbon will form a bond with an oxygen, and when it shares a bond with oxygen, the oxygen is greedy and will sort of hog the electrons that it's sharing with the carbon. So the carbon in each of these steps is losing its electrons to oxygen. When the methanol is converted to formaldehyde, another polar covalent bond forms between the carbon and the oxygen, and the carbon loses more electrons to the oxygen. And then the carbon loses more electrons in formic acid to another oxygen, until finally this carbon is fully oxidized. As carbon dioxide, the carbon has lost its electrons to each of these two oxygens. The 4 pairs of electrons it's sharing with those 2 oxygens spend more of their time around each of the oxygen than they do the carbon. So in this illustration, what we're showing is the stepwise oxidation of methane to carbon dioxide. Each of those steps is releasing some energy. Now, in the case of respiration, we will be following stepwise oxidation of sugars. Energy released from the sugars transferred to high energy carrier molecules. And along the way, we'll be watching the sugar molecules or the fuel molecules getting smaller, and we'll watch carbon hydrogen bonds being replaced with carbon oxygen bonds as the fuel, in this case, will be following glucose. The sugars are oxidized. So let's look at an example of a series of molecules, not really a series, but a set of molecules, and ask you here, if we look at, let's see, the first one is ethane, B is ethanol, C is oxalic acid, and D is carbon dioxide. Which of these molecules in this set is the most reduced? How about 5 more seconds? And we're really looking at the carbons, right? Which carbons are most reduced as opposed to which carbons are most oxidized. All right, let me close it up. And of all of these carbon-based molecules here, yes, the ethane is the most reduced molecule, right? If we look at each of those carbons, each of those carbons are sharing non-covalent bonds with hydrogens. So the carbon hasn't given up any of its electrons, as opposed to the most oxidized form of carbon in carbon dioxide. The carbon has lost its electrons to each of those oxygens. Right? So the ethane has more free energy the carbons have more free energy in ethane than the carbon does in carbon dioxide. So again, getting ourselves ready to follow cell respiration, the breakdown or the oxidation of sugar molecules, what we'll be seeing is the sugar becoming oxidized. The sugar is losing electrons. It's losing electrons in the form of hydrogen atoms. Hydrogen atom is an electron and a proton. Those electrons are donated to an electron carrier, and the electron carrier becomes reduced when it accepts those electrons. The main electron carrier that we'll be following in cell respiration is NAD+. Now, electrons are also moved around in photosynthesis, which we'll be talking about 2 weeks from now. And the figure that we have to show from our textbook is actually a related electron carrier called NADP+. In either case, the same kind of electron transfer occurs where the oxidized form of these molecules accepts 2 electrons and a proton, which we call a hydride ion, and they become reduced. So for example, in photosynthesis, the NADPH is the electron carrier that's reduced in photosynthesis. NADH, without the P, without the phosphate, is the electron carrier of respiration. So you can remember NADPHP for photosynthesis. So let's look at what do we mean by the difference between an oxidized form of an electron carrier and a reduced form of an electron carrier. Over here on the left-hand side, again, we're looking at NADP because there's a phosphate down here that's present in the NADP form that's used in photosynthesis. NAD plus is oxidized. And while I'm showing you the molecule so that we can understand what this molecule, where and how it's carrying electrons, I'm not asking you to memorize its structure, but what does NADP mean? N, it's a dinucleotide, that's what the D. One of the nucleotides consists of a nicotinamide ring. That's what this nitrogenous base is, attached to a ribose sugar, attached to a phosphate. So this is a nucleotide. It's attached via its phosphate to another nucleotide that we're familiar with, adenine. So NAD is a nicotinamide adenine dinucleotide. And sometimes it has an extra phosphate on it, so we call it NADP. Plus oxidized form because this nitrogen is missing an electron, so it's positively charged. This electron carrier can pick up electrons from food. 2 electrons plus a proton that we call a hydride ion, and the symbol is capital H with a negative charge, a hydride ion, are transferred to the nicotinamide ring of NAD or NADP to be reduced. One of the electrons will neutralize the positive charge on the nitrogen. One of the electrons and a proton in the form of a hydrogen atom will form a new bond with the carbon at this position on the ring. So again, I'm not asking you to show me the chemical conversion or the chemical change, but to help you see how NAD plus or NADP plus can carry electrons. They will carry electrons in this nitrogen and in this hydrogen that have been added in this reduced form of the electron carrier. So this is a high energy electron carrier because those electrons came from the high energy food molecule. And what we'll be seeing in both photosynthesis and cell respiration is how these electron carriers are sequentially oxidized and reduced, carrying electrons around from one molecule to another. And that's what we see here. Molecule 1 is oxidized when its electrons in its hydrogen atoms are transferred to the electron carrier, the oxidized form, and the electron carrier becomes reduced. It picks up 2 electrons and a proton, carrying it in the NADPH, and another proton from this original molecule is released into the solution. This reduced form of NADP, high energy electron carrier, can transfer those electrons to another molecule. And then the NADPH is oxidized back to the oxidized form, and the molecule that it transfers its electrons to becomes reduced. So electrons from one molecule transferred to the electron carrier, which can then later transfer it to another molecule. Transfer those high energy electrons to that molecule, reducing molecule 2. So we're not looking at any particular step right now. We're just thinking about conceptually what an electron carrier is doing, how it's carrying electrons around. So we've described both NADH and NADPH. These are the reduced forms. Activated as in high energy activated carrier molecules. One functions in cell respiration, the other functions in photosynthesis. But they both have similarities. They're both carrying what? And what do we call that? We call that a hydride ion. What is it that they're carrying? So think about, what is a hydride ion, right? What did I tell you is a hydride ion? 5 more seconds. Let me close it up. And D is the correct answer. When they're reduced, they're carrying 2 additional electrons and 1 proton. Together we call that a hydride ion. The reduced forms, that's what the H is, the reduced form of these electron carriers, NADH and NADPH, activated carrier molecules, high energy molecules carrying electrons that were extracted from the fuel molecules. And we have another activated carrier molecule that we'll be describing. Again, we don't need to memorize or be able to draw the structure, but notice this common theme of this molecule, acetyl-CoA, acetyl coenzyme A. It starts with, or part of it is another nucleotide, an adenine nucleotide with this very long carbon, nitrogen, oxygen chain. At the very end, we have this sulfur atom that can bind to an acetyl group. We'll see where that comes from later on, which actually came from the food in oxidized form of our sugar molecule, forms a high energy thioester linkage. This is another molecule that carries energy in the form of its chemical bond that is important for driving forward a non-spontaneous reaction in cell respiration. So when this thioester bond is cleaved and this acetyl-CoA is oxidized, it will release a large amount of energy that can be used to drive another reaction. So we're just introducing this concept of this molecule. This is not something I'd ask you to draw or be able to recognize, but we'll be talking about it in one of the steps of cell respiration later. So we have ATP, high energy molecule. We have reduced electron carriers, NADH. We have acetyl coenzyme A. All of these are helping to move this reaction forward, this catabolic reaction of breaking down a sugar molecule and generating a large amount of ATP for the cell. So respiration is what we're getting ourselves ready for. A catabolic breakdown series of reactions. We could be, if we talk, respiration involves breaking down fats and polypeptides and polysaccharides. Any of those have to be first converted to smaller subunits. We're usually going to follow, and we will be following, the breakdown specifically of glucose. And glucose will enter glycolysis, a specific series of reactions breaking down glucose. So again, energy can be harvested from any of our food, fuel, macromolecules. Proteins broken down to amino acids, polysaccharides broken down to simple sugars, fats broken down to their fatty acids, and glycerol. All of those can enter cell respiration, but we're following the breakdown of glucose specifically. And the breakdown of glucose, the oxidation of glucose, occurs in the cytoplasm. All cells, whether you are a prokaryote or a eukaryote, Glucose is a source of energy, can be broken down. It's oxidized to pyruvate. That process, 10 enzymatically catalyzed steps we're going to be learning about next week, actually on Monday. You will be expected to know all of the intermediates in those 10 steps. We'll go over the steps, the enzymes that catalyze, that will follow the structures. You won't be required to know the structures of the enzymes, but that will help you understand all of the steps along the way. Oxidizing glucose to pyruvate. In the process, a little bit of ATP is generated and some electrons are transferred to the electron carrier NADH. Pyruvate then, in a cell that is not, does not have any access to oxygen, the pyruvate will then be reduced in the process of fermentation. But in the most efficient form of cell respiration, aerobic respiration, when the cell does have access to oxygen, pyruvate will be further oxidized to acetyl-CoA. Acetyl-CoA, and again, all of this goes on in the cytoplasm. Acetyl-CoA can then be transported into the mitochondria. And in the mitochondria, it then enters the citric acid cycle, a cycle of biochemical changes that result in fully oxidizing the rest of the carbons to oxidize to carbon dioxide, which is then released from the mitochondria, released into the cell, and then diffuses out of the cell, and we breathe it out. Citric acid cycle generates more ATP, and citric acid cycle extracts more electrons, carrying the electrons and electron carriers, NADH, plus another similar one that we call flavin, adenine dinucleotide, FADH. And all of those electrons are carried to the electron transport chain. Again, in the mitochondria, where oxidative phosphorylation occurs, generating a huge amount of ATP, requires oxygen, which will be reduced to water. So we will be covering all of these stages of cell respiration, glycolysis, citric acid cycle, oxidative phosphorylation next week. And we can see them, again, sort of as an introduction of the glycolysis occurring in the cytoplasm, generating a little bit of ATP and a little bit of NADH. Pyruvate entering the mitochondria in white are double membrane mitochondria. Acetyl-CoA enters the citric acid cycle, generates a lot more NADH, and the electrons carried in the electron carriers will drive phosphorylation of ATP to make a huge amount of ATP. So that's a summary of everything that we'll be talking about next week. Now here we're looking at, this is a figure we had at the very beginning of this chapter, the chapter PowerPoints. Many, probably not all, many of the interconnected biochemical pathways of metabolism. And we've highlighted in pink here a central biochemical processes. And I'm asking you, based on what we just talked about, you didn't have to take biochemistry, what do you think lies at the center of all of these networks of reactions? Some are catabolic, some are anabolic, some are breakdown processes, some are building synthesis processes. What do you think is at the center of this? Which is why we'll be studying these processes next week. Everybody have a chance to vote? 5 more seconds. Let me close it up. A is the correct answer. The center of all of these processes, not letting me choose, there we go, glycolysis, breaking down glucose to pyruvate, pyruvate converted to acetyl-CoA that can enter the citric acid cycle. All cells, anything that's alive, can carry out glycolysis, oxidize glucose, at least partially. And cells that have access to oxygen, have a mitochondria, can carry out the citric acid cycle and continue to completely oxidize that glucose. And that's what we'll be talking about next week. So as a reminder of what you should hopefully know from this chapter, getting ourselves ready to think about energy processing in cells, what's the difference between a spontaneous and a non-spontaneous reaction? What's the activation energy? What is that barrier? And how can it be overcome? Why does the cell need ATP? Why does it go to all the trouble to make ATP in cell respiration? What's some of the jobs of ATP in a cell? What about all of the different chemical reactions of a cell? What do we mean by equilibrium point? How is it reached? Is it reached? And why does a cell oxidize sugars? Sugars as a fuel molecule. And how does it do that? Right. So think about those questions while you're studying, while you're preparing. Think about answering them as if they were essay questions, as a way for you to try to put together and synthesize all of the different concepts that we've been talking about. All right. So next week, start a new chapter. Some of it's in chapter 13, some is in 14, new PowerPoint. The last bit of material that we will cover in exam 2 next Monday, a week from Monday, covers chapter 9, 11, 12, 3, and then 13, 14 is cell respiration. All right. All right. See you guys on Monday. Have a good weekend.
Dr. Bear, I had a quick question about one of the slides.
Mm-hmm.
Like, like on slide number, I think like 37, the ammonia went to NH2. I don't really understand like why didn't just add NH3 to the molecule.